Hi, this is Professor John Galaska, and I'm going to do a little presentation for my research methods class regarding experiments, the four qualities that every experiment has, a true experiment, and then talk a little bit about within subjects and between subjects research designs. So, a true experiment has four qualities. One is a manipulation. Manipulation of the independent variable. And then we're going to measure changes, okay, either increases or decreases or whatever, in the dependent variable. We're going to compare the changes that we get, comparison, and we are also going to control for other variables so that they don't interfere with our manipulation. So at its very simplest, an experiment could be something like if we have a group of students and we feel that giving them a study guide is going to increase their performance on an exam, then we could say that the independent variable is the presence of a study guide or not having a study guide, and we would see then the group that has the study guide what the results were, which is the dependent variable. So the independent variable in this case would be the presence or absence of a study guide, or in many cases the presence or absence of a treatment. The dependent variable is going to be the scores on the exam, and we would see whether the group with the study guide got higher scores as we anticipated or not. We would compare those two and then make a conclusion. While we did this, we would have to control for other variables. In other words, the group that we gave the study guide to would have to be very similar to the group that did not get the study guide. And the conditions under which they took the exam would all have to be the same. For instance, it wouldn't be fair, or it would not be, there would be a confound if we were to give one group the test in the morning and one group the test in the evening. True experiment has manipulation of an independent variable. Then we measure what happens to the dependent variable. We compare those two things, and while we're doing this, we control for other variables such as environment and other conditions that might affect the dependent variable besides our independent variable. In other words, we want to isolate the independent variable as much as possible. All four of these things are present in a true experiment, and a true experiment is necessary for demonstrating causality, cause and effect. Okay, so we can demonstrate by the experiment that I just used as an example that the use of a study guide does or does not influence scores on an exam at the conclusion of the course or whenever. Okay, so these four things are part of a true experiment. Manipulating the independent variable, measuring the effects on the dependent variable, comparing those effects, and while we are doing this, controlling for extraneous variables so that we don't have something that contaminates the process. True experiment can demonstrate causality. It's pretty straightforward stuff. I'd like to talk a little bit about within subjects design and between subjects design for research. within subjects designed, each of these has unique advantages and disadvantages. Within subjects and between subjects. And when you are doing your journal critique, you should be able to identify whether their research design is a within subjects or a between subjects design. A within subjects design has one pool of participants, and everyone in the group gets treatment 1 and then gets treatment 2. Every single member of the group gets each of these treatments. And what happens when you are giving treatment 1 and 2 to one particular individual is you eliminate the individual differences in variability in subjects. In other words, since we are using person A, we know that person A is a set package of individual differences, a set package of personality and all the other characteristics that go with it. But that package stays the same during treatment 1 and treatment 2. Okay, so you could say that individual differences are not as crucial in a within subjects design. However, there is the possibility that something happens to this individual between treatment 1 and treatment 2, and that's what's known as history. Sometimes also order effects, because if you give treatment 1 and then give treatment 2, the results might not be the same as if you give treatment 2 first. But you have some kind of an effect over time. So history effects are increased. So an advantage is individual differences are diminished because we work with the same set of characteristics with each participant in treatment 1 and treatment 2. Now, between subjects design, you have two pools, or one pool that you've divided into two, and you take all of these people in this group and all of the people in this group, and you give them all treatment 1 here, and you give them all treatment 2 here. Now, obviously, we don't have the same situation regarding individual differences. It's possible that this group is different from this group. Individual differences can become problematic. Supposing for some reason when we selected our random samples, we somehow ended up with a sample that had more females in it and the other more males. So that might be a confound. So individual differences here can be problematic. Okay? However, the treatments are given at the same time to the different groups. So there is no history or order confound. So you can see that these two methods, these two research designs, sort of have meshing advantages and disadvantages. Within subjects design, you give treatment 1 and treatment 2 to each individual. Because of that, the individual differences between T1 and T2 for each subject are downplayed because you've used the same subject. However, there is the possibility that something happens between T1 and T2. Maybe this individual has a traumatic experience, or if there's a long amount of time, maybe they become more mature. At any rate, history and time can affect what happens between T1 and T2. But individual differences are not as big a problem. Between subjects, you give the tests, the treatment 1 and treatment 2, at the same time. There's no lapse. There's no effect from history. There's no time for that elapses to say that, well, this group over here matured more than this group. They both get the test at the same time, treatment 1 and treatment 2. However, it is possible, just by random chance, that we have gotten a group here that is somewhat different from the group over here. That's impossible up here because the same person gets T1 and T2. But here, this whole group could be somewhat different from this group, and therefore individual differences become problematic. However, history differences are not a problem at all. So those are the aspects of within subjects design and between subjects design, the advantages here and the advantage here. The advantage of within subjects, individual differences do not affect the outcome as much. And the advantage of between subjects is that history or time elapsed between treatments does not come into play. Okay, so if you wanted to make a little diagram for yourself, you might go ahead and do that. And I hope that helps to explain within subjects and between subjects research designs.